Probably most of you had, when you had biology, you heard about various kingdoms of life. And in I think probably the first classification that people came up with was, can I eat it and can it eat me? And that was probably good enough. So after that time, they decided, well, looks like there's two kinds of things in the world, things that walk around and things that just sit there. And so they have the plant kingdom and the animal kingdom. For a long time, it was really clear that a lot of the things that they had clumped together in the plant kingdom were not very closely related. And even some of the things that were stuffed in the animal kingdom were not very closely related to one another. So in the late 60s, a guy named Whitaker thought about it, and he came up with a completely new scheme that I'll show you his basic scheme in just a second here. I keep going the wrong way. So the idea of this tree of life probably started with Charles Darwin. He wrote in his little... In fact, this is the only illustration in his work there, this little thing, drawing of a branching pattern. And he puts up at the top, I think, and he tells about things. Now, I don't know if you can see that other tree of life very well. Yeah, that's as big as it gets. If you can tell what's on there, number one, you have excellent vision. And number two, you might notice something wrong or odd about it. Can anybody tell me something odd about that tree of life?
Is there a humanoid?
There's Charles Darwin up at the top there. Yeah, it's Charles, but okay, I'll tell you. All of this is animals.
Right.
The dominant life forms on the planet are here.
Right.
Then they're almost not mentioned on that particular tree of life. So I'm going to correct some of that omission and show you little things in a little bit. So this is, it would be so much better if that were bigger.
We can move the table.
Or everybody can move forward.
Yeah, I can move the table. Maybe if you sat there, we can move the table. You are limited by...
You'll want to focus if you can.
Okay. Here we have bacteria-like things. When Whitaker came up with his plan, all of the prokaryotic organisms, that is, organisms that don't have a nucleus, were clumped together in a group called the Monera. It was a few years later someone realized that some of those things that we've been calling bacteria were fundamentally different than all the bacteria. And in fact, when they looked at the ribosomal RNA of different kinds of organisms, they noticed that they fall into three distinct clusters. Now you say, well, what is so interesting about ribosomal RNA? Well, ribosomes are the little bodies that read the information to make proteins. If the ribosomal RNA changes very much, you don't make the proteins. You don't exist. So ribosomal RNA is a highly conserved substance in cells. Also, it's pretty easy to isolate ribosomes. When you grind up the cells in your centrifuge, there'll be a little band there that's the ribosome RNA that you want to look at. So what they discovered was that there's this group called what was called the Archaea Bacteria, and then the true bacteria, or Eubacteria, And then everything that had a nucleus all clustered together, which we call the eukaryote. And so the thing is that even at that point there where we had bacteria and then we had the kingdoms, plants, animals, and fungi, there was this large cluster of things here that were called protista, and they were a mix of all kinds of things that are not very closely related. It was really obvious from the very outset that this was not a very good taxonomic category, but it stayed that way for several years until we started looking at ultrastructure and chemical composition and things, and now we can look at DNA really easily. And we have this, the shrub of life. And you can see the largest print there are the names of the three dominant groups, the domains. They're now called domains. And this is in a state of flux. People are looking at different things and they're finding, oh, well, this actually should be here and these things should be together. So this is the most recent thing that I have until I fix one little part of it, where those things up there called the chromalveolata and the rosaria have now been unified into a single supergroup. So this scheme, and the one that's mentioned on your little handout there, has 5 supergroups within the eukaryotic organisms. The excavata, the rosaria, the chromalveolata, archaeplacida, and the uniconta. What happened to those kingdoms that we had in the previous one? Well, the fungus kingdom is here, the animal kingdom is there, and the plant kingdom right there. Everything else that's not in the Archaea Bacteria, everything else above this that's not a plant, animal, or fungus, was formerly placed in the protista. As you can see, they are all over the place in terms of their relationships. So our ultimate goal is to have a classification that's based on evolutionary relationships. Okay, so there's some complications. They're not pure lines of things. What you have there is some crossing over of lines because of this, a couple of factors. One is bacteria and archaea are able to steal DNA from other organisms, even if they're not related. So you can get, and in fact, this is why there are so many strains of bacteria now that are antibiotic resistant, is that they can get genes for antibiotic resistance from other organisms, other cells that are not related to them, and so that all of a sudden you've got a new combination of DNA there. Also, viruses can transfer genetic information from one organism to another. And this happens even, apparently, in eukaryotes, in the so-called higher organisms, that viruses, when they go into one thing and they take over and then they come out and go into another organism, sometimes they can take genetic information, and sometimes, although most viruses are pretty host-specific, they can change hosts somewhat. So there is a lot of complication about trying to figure out whose DNA is what and so on. Plus the fact that in your cells, you have things that used to be bacteria. The mitochondria that handle a lot of your energy budget formerly were free-living bacteria that got taken into another cell, and they ended up as part of those cells, and most of the things that have come along since then have mitochondria. In fact, we can trace evolution of individuals to an interesting extent by looking at mitochondrial DNA. Now, you got your mitochondria from your mother. So this is a line of inheritance, the mitochondrial DNA, through the female lines. Then there's another thing through the male when they're looking at sequences on the Y chromosome, which are found only in males. But that's there's all these interesting other things unrelated to what we're going to talk about. But the thing of genes going from one organism to another is called lateral or horizontal gene transfer. And the situation where mitochondria formed and also chloroplasts formed from cyanobacteria, this is due to endosymbiosis. Okay. So you're gonna see there's a yellow blob that's gonna move around as we talk about the different groups. First one is the Archaea, and these are formerly bacteria. They look just like bacteria. The thing is that they have something interesting about them that puts them closer to us than it does to actual bacteria, and that's the fact that many of the Archaea have introns in their genes. Now, a gene is a section of DNA that codes for a particular protein. Introns are little non-coding segments that are in there, and they have to be edited out before that protein can be made. We have them, other eukaryotes have them, very common, and Archaea have them. Almost no bacteria have any introns in their genetic information. When they first started finding that a very small percentage of our genes actually code for anything, they call the rest of it junk DNA. It turns out that junk, a lot of it is actually important. So that's why junk is in quotation marks there. Another thing about the Archaea is that they are anaerobic. They don't do well with oxygen around. And they have a cell wall composition that is fundamentally different than the cell wall composition of bacteria, which have something called peptidoglycans in their walls.
So before you go on,
yeah.
The name is important too, right?
The name means old, ancient. Yeah. There is the notion that maybe they came first, but it's probably not true.
Oh, I'll be darned.
So, in fact, if we go back to that, there's some question marks here that you might not see. First life, we don't know. And then Luca is the last universal common ancestor. So there was a break-off. The bacteria evolved in one direction, and the Archaea and the Eukarya went off. And actually, we think that the branch point of the Eukarya is closer to the Archaea. Who came first? Don't know. So, can anybody read that?
Yeah.
Okay. Well, these are some of the major groups of the Archaea. They typically live in extreme habitats. You see halophiles, the halogens, group 17, they're part of many salts. So things that have very high salt concentration, evaporation ponds, about the only thing that can live in some of those things are some of these Archaea. There's also the methane producing. Now, they live in your colon and in the guts of a lot of other organisms. The only organisms that we know of that can actually generate methane, CH4, are members of this group. These groups, there's actually several of them. And so when you make extra gas, then that's some Archaea that are in your gut doing that for you. And cows do it, and termites do it. And anyway, it's a very interesting greenhouse gas that's even more potent than CO2. Fortunately, a lot of it is not available until the tundra defrosts, and we then get tons of it released. I'm counting on being dead before that happens. Okay, anyway, then there's a group that like it really acid. I don't know if you can see, but that 0.7, in other words, a pH below 1. You have cells in your stomach that secrete hydrochloric acid that's a pH of about 1. It's immediately diluted by the other things in there, so that your stomach acidity is maybe 2 or 3. And you don't eat your stomach because it's lined with the slime coat of mucus. Those guys love acid. In fact, if it gets above 4, now 4, if you put pH 4 on a cotton shirt, you would probably eventually have a hole in that shirt. But they like it at 0.7. That's pretty acidic. Then there's the hyperthermophiles, like it really hot. Optimal temperature for one bunch is 80 degrees Celsius. If you were put in a spa, 80 degrees Celsius, it would basically cook you. Yeah, you can cook food at as low as 60 or 70 degrees.
We call you stew.
Huh? And here is the record holder for liking it hot. It dies when it starts getting cold. In other words, below 90 degrees Celsius. That's cold for it. Their preferred temperature is well above the boiling point of water. How do you get water above that temperature? Well, it has to be under pressure. Okay. Now we in the labs we have these things called autoclaves that we use to sterilize stuff. You could put these guys in the autoclave and it doesn't faze them. They love it. Anyway, so there are some pictures of Archaea and their habitats. You see the deep sea vent. It's way at the bottom of the ocean, and so the water pressure is extremely high. So that's how you can get temperatures of water that's way above the boiling point. And you see some things that look like bacterial cells. You cannot tell them apart by looking at them under the microscope. All right. And then in the middle, at the bottom, you see salt evaporation ponds that are kind of pinkish or reddish. That red color is due to the growth of Archaea in those very, very salty environments where they are just letting the water evaporate off and it gets more and more salty there. Okay, moving on to the bigger group of prokaryotes, the bacteria. Notice the little yellow blob is moved over there. This is an enormously large and diverse group of organisms. Okay, so they don't have the introns. Now, an interesting thing about them is their little flagella. You see these guys here? This is an actual flagellum, and this is a diagram of it. And the creationists like to claim that a bacterial flagellum proves that evolution couldn't have happened because it's too complicated to have evolved. But it turns out that if you take out one protein, the whole thing doesn't work. But it turns out that's not true. There's a whole range of different kinds of flagella that have different numbers of proteins. And if you take away almost all of them, what you have left is this little thing, this thing here, which in some bacteria is a drill that they use to drill a hole in another cell to attack it. And so what they've done is some of them have added on to that and have converted what was a drill into a locomotion device. That's the story of evolution, really, is that you have one thing and it can get repurposed to do all kinds of other things. And you don't hear much about this, except one thing. Once they had an argument debunked, they still use it because there's a lot of people out there that don't know any better, and they that sounds almost reasonable. And I have a movie.
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rotobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell surface, make up the three parts of the flagella. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane.
Hydrogen ions.
In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins.
So you get the idea. This is a very cool structure. Eukaryotic cells also have things called flagella, but they are completely different kind of structure. And there is no way that they had any evolutionary connection. But there is an interesting possibility. Okay, so some of the important things about what we use to tell bacteria apart. One of the first ones is Graham Stain, a Danish scientist by the name of Graham. Figured out that when you stain bacteria in a certain way, some of them end up purple and some of them end up pink. And the purple ones have very thick walls and the pink ones have thin walls. It turns out that a lot of, that's a big dividing line in the bacterial world. And then you get a size comparison there of some of the range of some of these things that we're talking about. There's also the shapes of the cells and how they cluster together. Yeah, cocci and bacilli and spirilla and spirochetes, all the different shapes there, and then a few variants of that. Okay, so in the bacteria, there's 5 major clades that I'm going to tell you about. It's actually, the classification is a lot more complicated than that, but I've just kind of got the 5 way I want it. Okay, so the first group is the proteobacteria, which is a very large group, and it's divided up. I guess that, fraternity, sorority folks or something got involved in this because they're all named after Greek letters. So you got your alpha, beta, gamma, delta, epsilon groups of the proteobacteria. All of the proteobacteria are gram-negative, have the thin walls, and usually have a slimy coat on the outside. Now, within one of these groups, can you see the blue name, Thiomargarita namibiensis? That means the sulfur pearl of Namibia, and it is a gigantic cell. There's a fruit fly. Now, fruit flies are not gigantic. A fruit fly is about that big. But most bacterial cells are about 1 million times smaller than that thing where the arrow is pointing. That is a single cell of Thiomargarita. And then there's 2 dead ones up there. So in most cases, eukaryotic cells are much larger than bacterial or prokaryotic cells. But in this case, let's say this table could be a bacterial cell and this room could be a typical eukaryotic cell, whereas a Thiomargarita would be about as big as the library plus the other library over there. Alpha group has 2 cool things in it. It has the bacteria that are now in your cells, the mitochondria. And it has these bacteria that live in the roots of legumes, and they do a very important thing. Maybe half of the nitrogen that gets into living things is done by fixing nitrogen by these bacteria. The rest of it is done mainly by a physical phenomenon. The atmosphere is mostly nitrogen, but you breathe it in, you breathe it out, you can't use it. How does it get made into something you can use? Well, these bacteria do it. They can take it apart and add other things to it. But then when lightning discharges, that energy tears all the molecules apart, and then they go back together in various ways, and sometimes they produce nitrates and nitrites and stuff, and you've got nitrogen fixation by lightning. The beta group, only thing about this that is very interesting is that gonorrhea is in there, and also bacterial meningitis is in there. The gamma group has several interesting organisms. There's the Thiomargarita and the Legionnaire's disease, and Vibrio, little comma-shaped cells that cause cholera. Then the Yersinia pestis, which was formerly named Pasteurella pestis after Pasteur, but it turns out that the Yersinia name had precedence. And then everybody's favorite, everybody has E. coli in them, Escherichia coli, living in their guts. So that's in that group. The Delta group has myxobacteria that form weird little fruiting bodies, but it also contains this, what I call the Tasmanian devil of the bacterial world. This is a little cell, the yellow one, it swims along and runs into another cell and drills a hole in it and goes inside and takes over and kills it. And in fact, I have... I couldn't find a movie of them swimming around and attacking a cell. I only have a movie of them after the cell. They've already gotten into the cell. I think that's probably oops, what happened?
Before we start the movie, we see a tiny black Vibrio predatory bacterial cell inside a bigger fluorescently labelled prey E. coli cell. The Vibrio invaded this cell, and as we start the movie, we see the Vibrio elongate bidirectionally, growing and using the insides of the prey cell. Then it septates unusually to give 5 progeny cells, and the E. coli goes dark as those progeny cells lyse it and escape and go on to attack more prey bacteria.
If you have a stomach ulcer, it's because you've got Helicobacter living in you. And for many years, it was thought that the reason that people got ulcers is they worried about this and this and this. And it turns out that if you don't have that bacterium in there, you're not going to get stomach ulcers. The problem is that it's a fairly common bacterium, so you do have the option of getting that if you want.
Well, how do you get it?
How you get it?
I know, so...
Listen, something can damage, if something damages the lining of your stomach so that they can get in there and set up, then they can start consuming your tissues and growing there and eventually make an ulcer and eat through your stomach. Yeah.
Well, I don't want it to happen.
Okay, don't do it. The next major group is the group called the Chlamydia. These are obligate intracellular parasites. They have to be inside of a living cell in order to reproduce. They have an infectious body called elementary bodies. There we see some of them on outside of red blood cells there. But those elementary bodies can't reproduce. But they go into a cell and they convert into something called a reticular body, which then divides and makes more of itself and harms the cells, and then releases a bunch more elementary bodies, which go and attack other cells. And it gets into bodies mainly through mucosal tissues, like in the eyes or in the genital area. And it's a very common. It's the world's most common sexually transmitted infection, and consequently the most common cause of blindness due to infections.
How long does it take?
I beg your pardon?
How long does it take from when the infection starts to work?
I don't know.
But isn't it easily treated?
Yeah, most of them are pretty amenable to antibiotic treatment so far. Okay, the next major group are the spirochetes. These have a helical form. Now, you'll see that there's a shape called a spirillum and a shape called a spirochete, and they're based on that root word, that spiral. They're not spirals. They're helices. If you have a spiral notebook, you don't have a spiral, you have a helix. That metal thing that is a helix. A spiral changes diameter. Okay, just a little geometry. Anyway, so these helical organisms have a band of proteins that runs the length of the cell, and it can cause the cell to change, shape, twist, and corkscrew through the environment. I have a movie of it doing that. So there are some spirochetes. I think this is the spirochete that causes Lyme disease. Although, honestly, I don't know how you can tell one from another. I mean, I can. But anyway, so they squiggle along due to that axial filament that runs the length of the cell.
Do they propel kind of like a snake? Or, how do they turn?
Like this. They just kind of...
I think attraction is how what it is.
Well, they're pushing against the water that they're in. Those...
Inclined flame.
Yeah, if you have a spiral notebook and you just start turning it, what happens to it? It goes out the other end of the thing,
right. But what propels the turning?
The turning, the contraction of that axial filament. It's a protein that can change length, and it causes the whole thing, because it's wound around there, it changes shape, and it causes the whole thing to move. Oh, and Lyme disease, characteristic symptom of that is the bullseye pattern at the site of infection. The gram-positive bacteria, very large group of organisms, and what's in there is anthrax and botulism and tetanus, and staph infections, staphylococcus, streptococcus, also mycoplasmas, which are very small cells that don't have a wall, the actinomycetes, which the word mycete, the root means fungus. These grow as filaments, and so the thing is that they're not related to fungi at all. They just grow as filaments. So that's why they got the name mycete, because people didn't know the difference between fungi and bacteria and any other little thing. But nevertheless, they typically found in soil all over the world, and they and other bacteria and fungi have been fighting one another for billions of years, and one of their weapons is antibiotics that they use against one another. And so we steal antibiotics from them. Of course, the first thing that we stole antibiotics from was a fungus, penicillium. But now we've figured out how we can change the configuration of those things and make new antibiotics from them and stuff. And so you see there's some examples of them. Now, up in the upper right hand, the Clostridium, you see those round things. When conditions are not favorable for growth, they can form endospores, which are extremely resistant to being killed. If you can things, you have to get the temperature at a certain point long enough to kill the endospores. Otherwise, you seal it up, and if you happen to have gotten some Clostridium botulinum in there, then they will emerge from their endospores and start making the botulinum toxin, which is probably the most poisonous substance we know about. Now, people do like to inject it in themselves, or somebody inject it into them. That Botox is the botulinum toxin, and what it does is it paralyzes muscles. It makes muscles not work. And there's some other things in this group that do that sort of thing. For example, tetanus makes another kind of muscle paralyzing agent. And when people got tetanus, it's called lockjaw. When I was a kid, my mom was a doctor, and so if I stepped on something, she always gave me a shot. I was always getting tetanus shots for stuff. And I hated getting shots. And so one day I was on one side of the table and she was on the other. And we were doing this little dance. And I said, I tell you what, if I get lockjaw, then you give me all the shots you want. Okay, lockjaw doesn't sound like such a bad thing. But the next step after that is your whole body locks up and you're in excruciating pain until you don't have any pain at all. And so, anyway, I got the shot. And the last group of prokaryotic organisms are the cyanobacteria. Used to be called the blue-green algae. And these are the organisms that, when they evolved, were the ones that first started producing oxygen. And some of them became endosymbionts and formed the chloroplasts in plants and in some other kinds of things, like the red algae, the green algae, the golden algae, all the algae. Now, and we know them from the fossil record. These are fossilized stromatolites from, I think, Australia, and this is from modern-day Australia. If you were to cut one of those, you can actually, they're kind of soft. You cut them, you see they got these lines in there, just like this, and you can actually make a thin section of the fossil and look at it under a microscope and see the cells that look just like the cells in modern-day stromatolites. And so these are from, several billion years ago, like 2.5 or so billion years ago. And when they started making oxygen, the first thing that happened was that the oceans rusted out. Because the oceans, we're pretty sure, were full of iron salts, and the water would have been bright green. And then oxygen in the water caused the iron to rust out, and we have these ancient deposits called banded iron ore, which are a result of cyanobacteria making oxygen and binding up with iron. Once most of the iron had fallen out of the ocean, then they started making it, and it started changing the oxygen content of the atmosphere so that it became a place where things that were not anaerobic could survive and things that were anaerobic couldn't. And so it opened up a whole world of new possibilities that have been filled now. All right, so there's a big yellow blob that is the eukarya, sometimes called the Eukaryota. And it's going to, there's 5 major groups, supergroups we're calling them. And we're going to start with the excavata. Now, a while back I gave a slideshow on parasites, and so some of these guys were featured in that slideshow. We have our Giardia, which is an obnoxious little beast that looks like it's smiling at you, but not friendly at all. It causes bad diarrhea. Then there's its cousin, Trichomonas, which is a sexually transmitted infection and is characterized by a, pungent odor. Strong. Imagine you have some shrimp salad that you forgot about and you left it out on the counter for about 3 weeks. Anyway, nice olfactory impression. Anyway, then we have little green guys that swim around, Euglena, and we've got a living, swimming Euglena here.
The video shows a typical Euglena. The Euglena does not have a rigid cell, referred to as a pellicule.
It's got flagella, and it can also change the shape of its body. It's got chloroplast. It can make its own food, but they can also extract food heterotrophically from the environment. Yeah, and their cell wall, they don't have a cell wall. They have an outer coating, which is some bands of protein. It's called a pellicle. And this is one of the things when I was taking biology. Well, is it a plant? Is it an animal? Well, I say, well, guess what? It's a human. Then there's Leishmania, which is over here in one of these groups. And Leishmaniasis, which is transmitted by a thing called a sandfly, a little biting midge-type critter. It's in Texas. Several years ago, somebody had this weird condition on his skin, and he goes to the doctor. The doctor said all of a sudden nothing they did to it made any difference. And we go from one doctor to another, and finally somebody that had had some experience with tropical medicine took a look at it and said, oh, well, it looks like Leishmaniasis. We're starting to have the opportunity to see some tropical diseases right here. We don't have to go anywhere to get them. We can get them right here at home. And another one in this category is, you see a little bug in the lower right-hand corner? It is actually a true bug, a triatoma. It's called a pissing bug. And they transmit trypanosomes, and you see next to it, there's some red blood cells, and then the little squiggly purple things are trypanosomes. And it causes Chagas disease. A cousin of it in Africa causes African sleeping sickness, and there it's transmitted by the infamous tsetse fly. But that's where those guys are found.
Are you just trying to depress us?
I find it interesting.
That we could all get bugs and die?
You know, that's always an option. You can do those things, or you know, you could walk out and, you could drive out in the street and have somebody not pay attention and run over you.
It happened.
Yeah.
That's the truth.
See? Well, at least you survived it. And you fell in the river or the lake, whatever it was. Somebody pulled you out. I mean, you have different ways that you can go. Or not. Okay. So anyway, there's this another little guy in the excavata group is Naegleria fowleri, which lives in the lakes around here. It is a common freshwater amoeba and ordinarily doesn't cause a problem. Now, the thing is that when water levels of lakes drop, they get concentrated. And people that, if you go swimming in it, there's a pretty good chance you will not get them. But if they get in your nose, they have the opportunity to then start growing and eat into your brain, and they will kill you. And somebody in Louisiana, there was apparently people use these things called neti pots, and they run water through the sinuses. It sounds crazy to me. Anyway, she did that, and apparently the tap water. And in fact, just about 3 weeks ago, I saw another report that a town in Louisiana also has Naegleria in its water supply. And so apparently what they're doing, treating the water, is not adequate to kill these organisms. They need to beef that up, I think. Anyway, and you see there they are on the right, little amoeba-looking critters. The next group, Rhizaria. Most people, you will probably never hear about these again, but they're very interesting little guys. And include the ones in the very middle are foraminifera. They are single-celled amoeba-like organisms that make a shell like a snail does. And so as they grow, the cell gets bigger and they make a bigger and bigger shell. And then there's some other things called radiolaria. Now, there is also this group of things called heliozoans, which are in here. And this, I'm going to show you this one clip here, just so that you realize that I'm not the most boring person in the world. Maybe. Here we go. What's going on?
You probably don't have an internet connection here.
I'm not supposed to have an internet connection.
Okay.
It's supposed to run without. Oh, I didn't do that. I went and got the clips and put them as little. I forgot to do this one. Anyway, there's a guy says, if you have a high-speed computer, you might want to go to full screen. This is a Heliozoa santanica, So that was an excellent impression of the guy that you missed hearing.
So are diatoms part of this?
Nope.
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 All right. Bad guess.
All right. Now, this is a very large, diverse group. The Chromalveolata was created by combining a group called Chromista with the alveolata, and now we have two major subgroups in the Chromalveolata. The Straminopiles, which we will talk about in a second, and this group, the Alveolata. Alveoli are, as you probably know, are these sacs in your lungs. This is a different type of sac. These are microscopic sacs under the plasma membrane, the cell membrane. So it has nothing to do with the things in your lungs. But these alveolates have those sacs uniquely under their outer membrane. In this group are, on the left, we see the apicomplexans, which is the group that contains malaria and toxoplasma. Toxoplasma is the little thing that gets in your brain and sits there and makes you less afraid of things that you should be afraid of. And it normally works between cats and rats. And when it gets into the rat brain, it makes them unafraid of cats. And they go out and get eaten by a cat. And then they actually do their sexual reproduction inside the cat. And the cat poops it out and the rat eventually gets it. Or it gets into to where sheep or pigs or something are, and then if you eat meat that's undercooked, you have the opportunity to get them in your brain. Or if you are very careless when you clean out the cat box or petting your cat, you can get them, and they advise women that are pregnant to not handle cats and cat boxes and stuff, because it doesn't hurt most people, but fetuses and people with HIV infections where their immune system is compromised, it will eat big chunks out of the brain and kill them. And that malaria, the mosquito and okay. And now, here is representative of a very large, diverse group called the Ciliates. And our example is this little slipper-shaped Paramecium. If you saw anything under a microscope in high school, you probably saw Paramecium. I mean, that's a common thing. And so they're covered with little hairs that move, little cilia that help them move around. And there's other kinds of ciliates that do different things with their cilia, like they will make currents in the water and bring food particles into where they can eat them. Now, this is a single cell, and they're
fairly
it
complex.
seems like most things, microscopic organisms, microbes, tend to like classical music.
They grow better with it too.
Yeah. And here are two ciliates. One is Paramecium and the other is Didymium, and the Didymium is eating the larger Paramecium. So those are both in the same group, and they respect no boundaries there. Okay.
Is that real time or
No, it was speeded up. A little bit speeded up. And then there are so we have the apicomplexans, malaria, toxoplasma, we have the ciliates, and then another pretty large group, the dinoflagellates. This is a shell, their outer casing of one. They have some interesting qualities.
Perhaps you've seen these videos online. They appear to show glowing blue waves breaking on the shores of some alien beach, but this is actually San Diego. To find out exactly what's going on here, we talked to marine biologist Michael Letts at the Scripps Institution of Oceanography at the University of California, San Diego.
So this video shows bioluminescence that is coming from a single-celled organism called Dinoflagellum. And in this case, there's really high concentrations of species, which is called Lingulodynium polyhedron.
According to Lax, this organism is known to form algal blooms commonly called red tides.
These red tides are just a natural part of the ecosystem, but the challenging thing about them is that we don't understand why they occur, and we can't predict when they're going to occur. We know that there's a red tide when we actually see it.
There you go. When you see it, it's there. Some of these organisms make toxins that are very irritating and actually can be harmful, particularly when ingested. And you say, well, why would you eat the red tide? Well, the thing is, oysters and other filter feeders, if there's a red tide bloom, and the oysters will pull in the water and filter out these organisms, and then if you eat the oysters, and I understand there are people that eat oysters, but that's a good way to get heavy metals and toxins and things from the environment, because they will take all that stuff and save it in their tissues.
Just for you.
Is there a good way to clean them? That doesn't happen.
Make sure that there's not a red tide, and within a few weeks of when you collect the oysters. In terms of cleaning the oysters, I can't imagine how you'll do that.
Okay.
Just don't eat them. That's the thing. Then the other major subgroup of the chromalveolators, the Straminophylla, they are the distinguishing feature of this group is something that not all of them have. But if they do have flagella, they will have one that's a normal kind of smooth flagellum, and another one that's got little tinsel on it, little appendages. Now, diatoms, here's where the diatoms are. We only know of one species of diatom that actually makes flagellated cells, and when it does, it makes them with that interesting little characteristic. Diatoms are algae that have silicon dioxide as their wall material, and they're like little petri dishes. And so when they divide, the two halves there's a cover and a bottom. So one fits in the other, and they come apart, and each half makes a new bottom. You say, well, how does that work? That was smaller than this. So yes, you have a smaller, and when they get so small they can't divide anymore, they have sex and start over again big. Also in that...
like reaching nirvana. It's like reaching nirvana.
The brown algae. Now, some brown algae are microscopic, but many of them, the ones that you may have encountered, are the kelps, giant kelps. Also, sargasso. The sargasso sea is this brown alga that there's hundreds and hundreds of square miles of it out in the North Atlantic in this big gyre out there. It's probably collecting some trash out there by now. But anyway, it's been out there millions of years, and there's a whole community of little organisms that live in it, and some of them are mimics of the sargassum. There's little shrimp and crabs and fish, anglerfish, and all kinds of things that look like the alga. And then there's the water mold group, the Oomycetes or Oomycota. This is the organism that killed off most of the potato crop in the 1840s in Ireland, Phytophthora infestans. Now, this used to be clumped in the fungus category, but it's not a fungus. It's fairly distant related. The main thing it has in common is that it grows as filaments, hyphae, and it kills off potato plants like crazy. And so back when that happened, about 1.5 million people starved to death, and another 1 million people moved, and a bunch of them ended up in Chicago, New York, and Boston. And Boston maintained a very Irish character, so that when they made a basketball team there, they named it after the Irish, the Boston Celtics or Celtics. Without that fungus-like critter, there probably wouldn't be a Boston Celtics. It would be the Boston something. Anyway, Oh, and I have a movie. Oh, I have a movie of Diatoms. They have a mysterious method of movement, this gliding motion, which is not understood. But they do it. It's not just the water moving them around. They are actually gliding. They think that some of them think that it's kind of like a squirting out some kind of goo out one end or something. Actually, we don't.
Brownian motion.
It's not Brownian motion. No, it's definitely not Brownian motion. They are moving in a direction.
Don't water skaters release some sort of?
Yeah, this thing that changes the surface tension. But these are in the water, so that the surface film has nothing to do with what they're doing. Anyway, it's an interesting thing. If you want a PhD project that lasts you about 40 years, then you got a good one there. Okay, and water moles. I have a movie of water moles when they're infecting something, and they get most of the food. They have to find new food. And so what they do is they produce these little zoospores. And you can't see the flagella on them, but on the side, it's kind of like a little bean-shaped thing. And on the side, it has these flagella, one of them smooth, one of them tinsel. And they swim around and find something. And if you want to catch some, what you do is you get a water sample and you put bait in it. And you bait it. What we would bait it with was hemp seeds. Hemp seeds were excellent attractors for water moles of various kinds. So we had a sack of hemp seeds in the lab. They were sterile hemp seeds, so you couldn't actually grow hemp plants with them, otherwise known as marijuana. You could also use, if you had some fruit flies around, you could sterilize them and put them in the water, and
So this is the type of reproduction I'm seeing? This is a type of reproduction?
This is asexual reproduction.
Okay.
They're
not dividing, huh?
They divided. They were inside this sporangium, and then they formed these little spores, and then they leave.
Okay.
And then they swim around until they find a food source, and then they start growing filaments, hyphae, growing into the food. See, fungi and fungus-like things grow through their food, and they secrete enzymes that break it down, and then they absorb it. Oh, so last one.
What's the process that causes them to? Here comes one more. What's the process that's causing everything to flow that way?
I don't know. That's a good question. It could be magic, but I don't know. That's always that's the advantage of if you're religious, then you can say that the zoospore moves in mysterious ways.
May I ask a question? With those spores and with the potato family, is there ways for that to have transferred to people?
Start over again. I missed the first.
With that particular cell and the potato family, is there any way that
they don't affect people. They kill the plant, which that was what affected the people. It was the fact that there were no potatoes to eat that killed the people. Yeah. The next to last of the supergroups, the Archaea, ancient plastid group. It has the plants in there, and it has the green algae and the red algae. Now, we think of plants as, well, they're really important on this planet. They are the primary source of food by taking energy from the sun and making it into chemical energy that we can use. But sometimes they do other things. They don't just sit there passively.
Fly traps. Did you ever imagine that a plant could devour such large creatures?
Poor little dog. Venus flytraps are native to North Carolina in the world, and now they are cultivated in other places. But They and other insect or frog catching things live where there is a low level of nitrogen in the soil, and that's how they fertilize themselves, is by catching and killing animals and letting them decay, and then they get the nitrogen compounds that way. Now the green algae, very closely related to the plants, they have the same kind of chlorophylls, they have the same storage product, starch, and they have the same cell wall material, cellulose. And here is Volvox. No, I thought I had music to this, but some Strauss,
right? Definitely.
Anyway, this is a colony of 512 cells, and then some of the cells are specialized to form new colonies of 512 cells. You say, well, why 512? Well, there's 1, and then 2, and then 4. You start doubling, and eventually you get to 512, and that's when they stop counting. All right, the last supergroup has 2 major branches. The Uniconta, which means one flagellum, consists of the Amoebazoa and the Opisthocanta, which means flagellum on the rear end. Okay, so these are mostly things that creep around by producing pseudopodia, like the amoeba there. And I do have a movie of the amoeba. Really cool photography, so you can see all the little junk inside the cells. So they creep around, and if there were a food particle around, they would send out the pseudopodia, surround it, and then that food particle, it could be a live critter or it could be something, some detritus, is now inside of a food vacuole inside the cell. And then it would secrete enzymes into the vacuole to break it down to feed on it. Now, also in this group are these things called slime molds. That was the group of organisms that I studied as a grad student and grew them. In fact, my first semester at UT in grad school, I found a new species of slime mold on the campus, and my professor was very excited about that. So, anyway.
Congratulations, that's awesome.
Yeah. But the thing about slime molds is that, so they make spores, and the spores germinate and make a little amoeba, or they make something called a swarm cell, which is a flagellated cell. If there's enough water, they will make swarm cells. If there's not enough water, just a very moist coating, they will be these amoeba-like things. And they can switch back and forth by just changing the amount of water that's available. Then they have this habit of having an orgy, where you have a whole gang of them get together, and they're sitting there doing this, like this, and then 2 of them get particularly friendly and make 1 cell out of 2, and then the nuclei fuse, and then you have the start of a plasmodium, because then the nucleus starts dividing, but they don't divide into cells, and it makes this gigantic protoplasmic mass. Anybody see the movie The Blob? Steve McQueen's first starring role, it was the slime mold from outer space, and it would get bigger, and it had that quality of kind of pulsating, but I'm not seeing that in actual plasmodia. But anyway, it's this big slimy thing, and it ends up with little veins. What's happening here? This is a plasmodium, and it's moving in this direction. It's sending out little pseudopodia and creeping, and if it runs into food, it makes food vacuoles out of it. But in those veins back behind it, it's not individual cells. It's all one mass of protoplasm. And inside that, we see this Can you see the movement?
That is so cool.
It's flowing through those veins. And then it does that for a while. And then it slows down. And then it slows down and stops and goes back the other way.
Sorry, what is exactly?
The side of the protoplasm. It's a gigantic amoeba. It is hundreds and hundreds of nuclei and mitochondria and food vacuoles and everything that's in a cell squishing through those veins.
How big is it?
So I don't see any movement here.
Oh, well,
you'd have to be a little closer.
You can see it. I can see it. Maybe if you come up here, you can see it. Come on, hurry before this stops. Can you see it now? Yeah. See, now, this is why I didn't want you sitting back there.
Yeah.
Is there why does it go back and forth? What's going on?
That's a good question that I also don't know the answer to. Although I do know that there is a protein in there that is very similar to the protein that makes up our muscles. It's called myxomycin. So there's this interesting contracting protein. Exactly how that creates this flow is not understood by me, and probably I don't think it's by anybody, but anyway. So there's some mysteries out there that haven't been solved. And so after they do that, they feed on things, and then they form little fruity sporangia and stuff. Yes?
Is there any evidence that cells are capable